This is a G-protein coupled receptor transduction pathway, specifically an epinephrine transduction pathway. It begins when epinephrine binds with an epinephrine receptor and initiates a conformational change in the receptor, which allows a G-protein inside the cell to bind with the intracellular portion of the epinephrine receptor after the conformational change. This allows the G protein to drop a GDP molecule and pick up a GTP molecule. The protein then moves to an enzyme called adenylyl cyclase and phosphorylates it, which allows it to turn ATP into cyclic AMP. Cyclic AMP acts as a secondary messenger in this transduction pathway, which phosphorylates kinases. These kinases then phosphorylate exponentially increasing numbers of other kinases in what is known as a phosphorylation cascade. At the end of this phosphorylation cascade, kinases activate an enzyme that catalyzes the breakdown of glycogen into glucose, and then the glucose is released into the bloodstream.
In the ion channel signaling pathway, the ligand binds to a membrane receptor that can either open or close to allow or block the diffusion of ions across the plasma membrane. The change in ion concentration leads to the activation of enzymes, which can activate second messengers who then work in the nucleus to carry out a cellular response. For example, if the ligand capsaicin is bound to TRPV1, a membrane receptor protein, the gate will open, allowing calcium ions to diffuse across the membrane, resulting in an influx of calcium concentration inside the cell. That influx activates calcineurin, which dephosphorylates the second messenger, NAFT2. NAFT2 enters the nucleus and begins inhibiting proteins like AFT3, leading to apoptosis as the final cellular response to capsaicin. In RTK signaling, when a ligand binds to the ligand binding sites on the two inactive tyrosine receptor protein monomers, they close together. Then phosphates are transferred from ATP to the tyrosines on the receptor to fully activate the RTK proteins. The active RTK then activates relay proteins, which then phosphorylates more proteins using ATP, starting a cascade to activate more proteins. The ending protein then enters the nuclear membrane to trigger a cellular response. In the case of the ligand, the epidermal growth factor, or EGF, when it binds to the EGF receptor, it activates the RAF protein, which then activates MAPK kinase, who then activates MAP kinase. MAP kinase then enters the nucleus to phosphorylate transcription factors, leading to the transcription of genes promoting cell growth and division.
This is an intracellular signaling transduction pathway, specifically an aldosterone signaling transduction pathway. To start off, aldosterone diffuses through the cell membrane of the capillaries. Once inside the cell, aldosterone binds to the mineralocorticoid receptor. This hormone receptor complex then diffuses into the nucleus, where it binds to specific genes and acts as a transcription factor. Once transcribed into mRNA, the mRNA exits the nucleus and is translated by ribosomes into specific proteins. The proteins it is translated into are the epithelial sodium chloride channel and the sodium-potassium ATPase pump. These allow for potassium to exit the cell into the tubular lumen and for sodium to be taken up from the tubular lumen into the cell.